Assalamu alaikum. Hello guys, welcome back to my channel. Today I'll explain about one of the most common diseases in my playlist of cow, of ruminants and domestic animals, mammal domestic animals. We have mastitis. Mastitis is a disease known as the inflammation of the milk gland. So the inflammation of the milk gland is called mastitis. Okay. We have a lot of causative agents. The causative agent may be sometimes viral, but not that common. We have, for example, cowpox. You will see, for example, lesions on the outer skin of the mammalian gland, but mostly bacteria and fungi will cause the disease. Causative agents are Mycobacterium tuberculosis, that doesn't have any treatment at all, and Candida albicans as an example of yeast, okay, Streptococcus agalactia and Staphylococcus aureus, and you have some other types of some other causative agents that I will explain about, okay. In cases of unknown causative agent, culture is the best choice. So TB, a note, it's important to know, TB tuberculosis can't be cultured easily, or it can't be cultured. Culture isn't done. We do other tests that I'll explain in other videos. Here is an example of cow mastitis milk. Pus will be seen in a milk. You see, as the milk colors clearly know that this is an abnormal color of a milk, as you can see, okay. Quickly, let's go to gangrenous mastitis. So what's gangrenous mastitis? It's gangrene. It's gangrene of the udder, okay? Gangrenous mastitis is generally caused by the bacteria Staphylococci, okay? We have staph bacteria, as I say. Secondary invasion is by Clostridium species and E. coli. So what type of Clostridium? Mostly Clostridium preferens, okay? Preferens, okay? And they are acute. And gangrenous mastitis are acute, with symptoms of, we will have pain, fever, blue or black discoloration of the udder, necrosis, which is caused by the alpha toxin. I didn't write, sorry. Let's correct this. Okay, it's caused by the alpha toxin, and fluid exudation. So it will appear just like edema. Here we have a picture. As you see, these two quarters are turned into black color. So this is a gangrenous mastitis, and gangrenous mastitis is painful when you touch it, okay. Culturing method. So how is culturing done? We take milk from the infected quarter aseptically. So what's aseptic technique of sampling? So aseptically, I've done this, I think, about 10, more than hundreds of times I've done this technique. You will bring a clean, sterile tube. Be careful. Clean, not only clean, sterile. Sterile means it's free from any pathogens. It doesn't have any bacteria, even one single cell, okay, of bacteria. First, clean, sterile tube. You will wear gloves. You will clean the udder, okay, clean the udder by, first I refer washing the udder by only water, and second you dry it. After that you can use chlorhexidine or iodine. I will use iodine personally. Iodine is the best, and you have iodine with you for in most cases. I will use iodine to clean the udder, and you will not touch the udder after using iodine, okay. So you will take the milk and put it in the tube. You will milk the cow. The milk should not touch your hand or any other surface, and you should put it directly in the tube. So the udder should be pointed to the tube at an angle, and you squeeze and milk the cow and put the milk in the sterile tube and close the sterile tube container, okay. You should close the cap and send it to a lab for culture. So what's culture? A swab will be used, a sterile swab will be used, sterile swab. For culturing, everything should be sterile. Of course, we'll prepare a medium for it. Which type of media is used? The sample is aseptically collected, as I said, from disease dose and submitted to microbiological culture on 5% debrinated sheep blood agar. So we will use blood agar and McConkey agar with the subword agar, okay? So McConkey is for only gram negatives, and we know blood agar is for gram negative and positive. This is the result you will get that I will explain about them videos. The results of bacterial cultures, okay? We have some histological appearance, okay? If we come to normal histological appearance, okay? The anatomy of the milk gland first. We have the opening of the lactiferous ducts. We have the gland sinus, okay, the milk glands, and we have the annular fold here. We have the teat sinus at the end. The milk comes out. When you squeeze the teats, we know that oxytocin is released, of course. That's an important point to know, that which makes the milk to get out, okay? Here we have a histology section. If we see a lobe of, as it's circled here, a lobe, this is a lobule, okay? Lobule of breast tissue, yeah. As you see, we have two ducts, intralobular duct and interlobular duct. So what's intralobular duct? The intralobular duct is, the intralobular duct are in the lobule. So this is a lobule, but the interlobular duct, as you see here, they are both lined by epithelium. It's out, okay? So the milk is released from the intralobular duct to the interlobular duct, okay? Here we have alveolus, those circular empty with empty lumen. Those are called alveolus, okay? They are again lined by epithelium. Those are called alveolus. So this was an H&D stain. If you magnify higher, if you get to a higher magnification, here we have the connective tissue, as I wrote here. This arrow that mentions here, this is called the myoepithelial layer, okay? And this is an example of alveoli of a milk gland alveolus at a higher magnification, okay? We will come to abnormal. So we know that mastitis is a type of inflammation. So what will we see here? What are we seeing here? We can see some couples of inflammatory cells. So when inflammation occurs in your body will white blood cells will migrate to the location of the inflammation to kill the pathogen. Okay, if it's a bacteria or any fungal or any type of infection. Okay, here I can see personally some couples of neutrophils. So the color is light, sorry. Okay. You can see some inflammatory cells, so much, not just some. I can see neutrophils here. Yeah, those are all neutrophils. This is an inflammatory cell. This is an inflammatory cell. So there are so much inflammatory cells here. This is the alveolus, okay? So this inflammatory cell indicates inflammation, okay? Those are leukocytes. If it's presented in a slide, that indicates inflammation. There is another case that's not something common. Thrombosis. Thrombosis may occur in gangrene. This is not something very common. Here is a venous thrombosis. As you see, a vein should be empty, okay, or there should be a little bit of blood cells. As you see, here we have a normal vessel here, but here we have a thrombosis. So if you come here, this is a thrombosis, and again we have here a thrombosis here too, as you see. So it's just like a blood clot in the vein. This is abnormal, and I will explain about thrombosis in other videos in details, okay? So treatment for mastitis. Mastitis, we have two types of mastitis, acute and chronic. Chronic mastitis, fever isn't seen, okay, in chronic mastitis. But if we come to acute mastitis, fever will be seen, acute gangrenous mastitis. And we have another types of mastitis, such as coliform mastitis, that I will explain about in other parts of the videos. So thank you guys, hope you get benefit.